With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello everyone, let's solve the question. Which one of the following statement is wrong with respect to RMS, that is restriction modification system, as the foundation of rDNA technology? Now, the restriction modification system is present in the bacteria to protect its own DNA from being cut by the restriction endonuclease enzyme, as well as to protect its DNA from a foreign DNA, for example, of a bacteriophage. Now, bacteriophage are what? These are the viruses that infect the bacteria, right? And they enter the bacterial genetic machinery and replicate, right? So to protect a bacterial cell from a bacteriophage or a foreign DNA, as well as to protect it from restriction endonuclease, bacteria have developed this system called as the restriction modification system. All right. Now this restriction modification system consists of two main enzymes. All right. The first is called as the restriction endonuclease. All right. What this restriction endonuclease does is that it recognizes specific sequences on the DNA strand of the bacteria as well as the foreign DNA, and then cut it on those sites. So these sites are called as the recognition sites. All right. So the restriction endonuclease will cut the specific sequences or these recognition sites. All right. And these sites can also be the palindromic sequences. All right. What are palindromic sequences? Which can be read forward and in backward direction, and they will still read the same. So these are called as the palindromic sequences, right? Now, the restriction endonuclease can cut at the specific sequences or the palindromic sequences in the double strands. All right. So suppose it is cutting here. So the sequences or the base pairs present here will be the in palindrome. That is, if they are read from 5' to 3' and on this complementary from 5' to 3', it will still read the same. All right. And the second enzyme present in the restriction modification system is called as the modification enzyme. All right. The function of this modification enzyme is to add methyl group at the ends cut by the restriction endonuclease. When the methyl groups are added to those ends, any foreign DNA cannot bind, all right, to those ends. All right. So this is how the bacterial DNA is protected from the foreign DNA being bind to it and then taking over its genetic machinery. All right. So let's see the options that are given to us here. The first one says restriction endonuclease identifies introduced foreign DNA and cuts it into pieces. All right. So this statement will be wrong. The restriction endonuclease does cut the foreign DNA but not into pieces. It will only recognize the specific sites and will only cut at those recognition sites. All right. So this statement is wrong. The second statement says modification enzymes add a methyl group to one or two bases, usually within the recognition site of the restriction endonuclease enzyme. So this statement is also correct. This statement is already right? Because we have seen that the modification enzymes add the methyl group within the this recognition sites. All right. So for example, this is the DNA strand and the restriction endonuclease has recognized this sequence. All right. So the modification enzyme will be adding the methyl group to one or two bases within this recognition site. Alright, so this statement is correct. Then restriction endonuclease can cleave any palindromic sequence. So this we have again seen here that restriction endonuclease can cut the palindromic sequences, right? So this will also be correct. And the last one says restriction enzymes restrict the propagation of foreign DNA of bacteriophages in the host bacterium. So this is what the function of restriction modification system is. So this statement is also correct. The only wrong statement is option 1. That's the answer. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.